us after that. Okay, so it's okay, right? Andreas, do you copy me?
Yep, that's fine with me.
Great. Okay, I thought your video is lagging because you seems that you're not moving. Sorry. Hi. Okay, great. We will wait in the next 2 minutes. Thank you.
So how many people have joined so far?
So far is 89 actually, and yeah, increasing because yeah, maybe the difference of time and people.
Oh, yeah, that's normal.
Yep. Cool. Based on the registration, there's a there are several people come from actually Java Island. Java Island. So Java Island and then Papua. Do you know Papua, Andreas?
Yes.
Yes, some people also come from Papua, and also Dipti. Dipti, maybe you remember Dipti. Yeah, Dipti will join us, and she already said from in the Twitter that okay, so you'll join us soon. I don't know if Dipti is in here at the moment. And yeah, we have also Miss Yulia Jabir, our Vice Dean. So
hello, Andreas.
Hello.
Yeah.
So she is our Vice Dean. Everyone seems so young. I mean, as you know, Mr. Subhan and Miss Yulia. So yeah, we are in the like young era of the dean and vice deans. Oh, Dipti hasn't joined us yet, so okay. Maybe it's time to start. We have the number are increasing, like 100, more than 100 now, and still going on. Based on the registration, we have 400 and around 490 people that's registered themselves to this webinar. And some of them, as I say, come from Java Island and Kalimantan Island, Papua, and Sumatra Island. So yeah, it's quite diverse. Okay, shall I start, Panure? Saya yang mulai deh, Panure?
Yes.
Okay, let's start now. Assalamualaikum warahmatullahi wabarakatuh. Good morning for Andreas and good afternoon for us in Indonesia. So we will soon start the webinar of the Faculty of Pharmacy number 2, because the first one is last month, and then this month we will start the second one with Professor Andreas Prokop, who has give us his time to deliver the presentation in neuroscience topic. And before we hear Professor Andreas talk, we will have opening remarks by Subhan, PhD, as the Dean of the Faculty of Pharmacy. Mr. Subhan, the time and floor is yours.
Okay, thank you, Dr. Visan. Our Honorable Professor Andreas Prokop, the University of Manchester, UK. Nice to meet you as our speaker today, and to all of participants. Good afternoon, ladies and gentlemen. On behalf of Faculty of Pharmacy, Hasanuddin University, I would like to thanks to our speakers today. We are really appreciated for your time to share of your research. I hope we can make a research collaboration between our institution in the near future. And also to all of participants, I hope you can learn and maybe you can find a possibility to do research collaborations and share of your idea or experience for to the better research. And recently, mostly of our lecture and seminar presented in online science of this pandemic of COVID-19. At least we can reduce the distance and then we can make a meeting in online. And this is maybe one of the new normal in this time. And then I apologize for maybe I cannot join until the end of this webinar because I have another meeting in another room. So I hope all will be enjoy with this seminar. And then again, thank you very much, Professor Andreas Prokop. Thank you very much.
Welcome.
And have a nice seminar. Thank you, Dr. Visan.
Okay, thank you very much, Dr. Subhan, for the opening remarks, and then we'll soon move to the main event of this of today is the talk by Professor Andreas Prokop. But before that, I would like to show you a little bit about the CV of Professor Andreas. So I will share screen to promote the website of the laboratory of where Professor Andreas belong. So this is the website of Professor Andreas Prokop team in Manchester University, in the University of Manchester, sorry. And then as you can see in here, the credentials, his credentials is really remarkable. And then as you can see in here, Professor Andreas graduated from Germany institutes. Following that, he did postdocs in the University of Cambridge. Before moving to Manchester, UK for his position as a senior lecturer at the Faculty of Life Sciences and become the professor for cellular and developmental neurobiology at the Faculty of Life Sciences, the University of Manchester in 2014. So it's been 6 years, isn't it, Andreas? Yeah, and as you can try to browse the website to find the possibilities to do a master or PhD in his lab. I'm sure he's welcoming people, probably. Maybe he can say that by himself later. Okay. Just short introduction that Professor Andreas Prokop actually with me tried to establish Drosophila community in Indonesia by using this. We call it Fly Indonesia, and then we have websites. Maybe Professor Andreas will promote it in the end. But yeah, feel welcome to ask about this once the session is over. Okay, great. So let's have your talk, Andreas.
Thank you very much. Good, so I will share my screen. So thank you very much, Birzan, for the introduction. And so can everybody see my screen now?
Yes, can see.
Good.
Your
So
screen.
I put this in the corner so I can see that everybody is still live. So yeah, merhaba. Assalamu alaikum.
Waalaikumsalam.
Inshallah, there won't be problems, technical problems. So let's do our best. And yeah, so what I would like to talk to you about today is our work in understanding an important aspect of the nervous system, which is how we maintain our nerve cells for many decades in our bodies, and how we can use, surprisingly, fruit flies in order to understand this and develop concepts that are then applying to medical conditions. So let's start first. What is an axon? So if you think of our nerve cells, then some of our nerve cells, like primary and secondary or upper and lower motor neurons, send cable-like processes, which are up to 1 meter long, but only up to 15 micrometers in diameter. These axons have to survive for basically 100 years if we grow old. which is, of course, an enormous challenge for such a delicate structure. Now, if we look at an axon, there is a highly specialized structure which contains many different organelles, and I don't want to go into any detail here. But what I would like to focus on are the microtubules. The microtubules are these filamentous proteins here, shown in green, which are forming a kind of backbone that run all along the axon. So shown here again in a slightly 3D view. And if we now take a diameter of an axon, as I'm indicating here, and we look at that in vertebrates, like for example here, small rat axons, then you see that the microtubules are fairly evenly distributed through the diameter of the axon. If we now go to an arthropod, so a spider or a crustacean or a fly, then you see that the organization of microtubules is very similar. And it is not only in the small axons, but also in large axons, you find evenly distributed microtubules, which is a picture very similar to what we see here in vertebrates. Now, people would say, well, that's all fine, but if you then look at larger vertebrate axons, then you will find, as shown here in magenta, many neurofilaments, so that microtubules are actually diluted into a sea of microtubules, of neurofilaments, which is a very different situation. Now, as you can read in this review here, I've looked into this and into literature of the last 50 to 60 years at the electron microscopy level. And basically, there's many conditions where you can take away neurofilaments completely or intoxinate axons so that you get a redistribution of neurofilaments into the periphery and microtubules in the center. What you then find is that the organization is very similar to a large axon in an arthropod. And the situation, if you take away neurofilaments, is not even lethal. There's a human condition, there are about 5 mouse models, and there is a quail model, all of which are viable. So we believe it's really these loose bundles of microtubules that are the actual essential core structure of an axon. Now, if we look at microtubules, then these bundles are important at any life stage of an axon. So if you have the early growth of an axon in the embryo or in regeneration, if you have branching effects during learning and memory, if you think of that contacts are established in the embryo, but then we grow to the full person size, then there's a lot of intercalative growth. Throughout all this time, we have microtubules as an essential factor. If you look at aging, then it has been shown that mammals, and this includes clearly humans, we lose about 40% when reaching 80–90% of our lifespan. So we lose about 40% of axons. And also in this context, it seems that microtubules may be very important. At least it has been reported that in aging axons or in neurodegeneration, microtubules become disorganized and form these kind of curled structures as shown here. And one of the questions that we are asking ourselves is, could this be a cause, or is this a mere consequence of axon degeneration? So what is the role of microtubules here? And in order to address this point, of course, we should first briefly go into how microtubules are regulated. So microtubules are protein polymers, and these polymers undergo something which we call dynamic instability. So as you can see in this growth cone, you see all the time microtubules advancing and then collapsing back from the periphery. And the process that underlies this is a plus-end polymerization and depolymerization of microtubules. So this is the fundamental dynamics we can observe in vitro, which also happens in vivo. But of course, in vivo, so in a living cell, this is not left to chance, but we are having many different proteins, and some examples are given here, which are binding to microtubules and which can regulate their dynamics. So we have, for example, cross-linking factors and stabilizing factors, factors binding to the polymerizing or depolymerizing tip. We have motor proteins which can run along these microtubules, other proteins which can sever and cut them, and others which can post-translationally modify these microtubules. Now, interestingly, and these are data from back in 2013 when I went to the OMIM database to ask how many cytoskeletal associated proteins are linking to human disease. At that point, it was about 60% of about 220 of those proteins are looked at, and out of those, more than 50% linked to neuro diseases. So this obviously shows that looking into these proteins may be of very high relevance. However, there's a problem, and the problem is that most of the work that is being carried out is basically we take a gene, we know it's linked to a neuronal disease, we then study its function. But from all this work that has been undertaken, we have not yet really gained a proper understanding of axon biology and the disease symptoms linked to these factors. The question is why? And the answer to that comes with a highly complicated slide, which deliberately is there to show you what reality is, or let's say an image of reality is, that we have many, many proteins which are acting on microtubules in parallel. And of course, we have to understand these networks and the underlying networks of protein function and not the single protein function alone in order to understand at the cellular level how the biology of an axon is regulated. Now, of course, this seems to be a daunting task, and the question is, how can we decipher such complexity? And our approach to this is to use Drosophila. So let me first introduce you a little bit to Drosophila biology and why we use Drosophila, then to come to the specifics of Drosophila in this context, and then to give you some examples, and then to deduce what kind of fundamental concepts we can come up with. So first of all, we start with this citation by Hugo Bellin. You get 10 times more biology for $1 invested in flies than you get in mice. And biology, I would add, is the lifeblood for translational research into disease. First, we have to understand the fundamental biology before we can think about the diseases which are related to this. So if we think of a fly, you have a life cycle of only 10 days, which makes our research very speedy. If we have to make genetic crosses, this is not an important time factor. If we think of raising and keeping flies, I would say we can keep 400 fly stocks, different fly stocks in a single incubator. That costs me 100 pounds per month. If I would keep the same amount of mice research ready, this would cost me in Manchester 12,000 pounds, or in excess of 12,000 pounds. So basically my cost savings are 100%. If you then think of genetic possibilities in fly, this is extremely straightforward, and there's many, many tricks elaborated. If you think of modern gene technology like CRISPR, fly is always at the forefront of technique development. So we have all these things that enhances our possibilities further. If we think of transgenic animals, to make a transgenic animal takes about 2 to 3 months, and it costs me perhaps 250 pounds. All this can be actually sent off to companies and done for me. It's very straightforward. Microscopy is very easy. I can put a whole embryo, which is just 0.5 millimeter in size, under the microscope and see the whole and entire animal. Actually, many of them under the slide, where in a mouse I would have to basically navigate across a section and only a small part of the animal. We have fantastic databases like Flybase, which is the definitely world-leading database in precision and detail. We have stock centers. So if I want to work on a gene, I very often just have to write to a stock center and they send me genetic tools for this strain. There is detailed knowledge and there's a tradition of sharing. So usually you write to somebody and say, you published this line, send it to me, people will send it. There's very rarely an issue. We don't need animal licenses. And in consequence, I always say that when we have an experimental idea, for example, over lunch in the laboratory, we either have the flies already or get them in. We do our experiments then a few weeks later and get our results, where in a mouse, you very often would first have to ask yourself, do we need a new animal license in order to perform these experiments? So I think there's huge advantages, but you may still ask, well, why is this important? I can do fast research, but why is this important? And the importance lies with the fact, and this is something, a statement by William Blake from 1794, am not I a fly like thee, or art not thou a man like me? So we have this concept of deep homology. And although we go back almost 500 million years to our last common ancestor, it seems that already this common ancestor had developed so fundamental biology that has been carried on into these different lines of animal kingdom, so that about 70% of disease-relevant genes in humans can be studied in the fly. And this has led so far to 10 Nobel laureates in physiology and medicine. So, of course, Drosophila is not a mini-human. This always needs to be stated. So I'll give you one example. If you think of Alzheimer's disease, yes, there are Alzheimer's disease models in fly. But of course, you cannot study personality loss in a fly. But what you can study is why nerve cells die. And these are questions that haven't been answered proper for the last 50 or 60 years. In fly, we can make advance, and you will see that in my talk, fundamental questions may come even from our research, or fundamental answers. Good, so I would therefore say that concepts of fundamental biology can be established in invertebrate models to inform then experiments in higher organisms. And this is really the pathway that we usually take. We work out the detail, the concept in the fly, which is fast, cheap, and easy. And once we've understood something, we then collaborate with other people in the field in order to test our results, which so far had surprising success. Good, so now let's come to the specific advantages for studies of axonal microtubules. So if you think of microtubule regulators, then they are highly conserved at the molecular level. If you see here, for example, these are different microtubule binding proteins or associating proteins and their distribution in axons in fly. And these are taken from the literature of the homologous proteins in different vertebrate organisms. You can already see there's many similarities in how these proteins behave. We have a good homology in our system. It's fundamental biology. Sorry, went the wrong direction. Second, of course, we have efficient genetics. So just to give you an example, we in our group, and I don't have a large group, I have a group of sometimes 7, 8, sometimes 3 people undulating over the years. And we looked over the last 10 years at over 50 actin and microtubule associating and regulating proteins, which is a unique database in one single system, cellular system. We can look not only at these microtubule regulators and actin regulators, we can look at all other aspects of axon biology, because for all these aspects, there's plenty of tools and insights already available. So I can, if I want to look at transport, very easily look at transport. If I want to look at mitochondria, I can do this very easy in the fly. Finally, this is an embryo of the fly. I can look at the embryo as a whole mount. It's just 0.5 millimeter long. We can dissect them. There are strategies to do this, but we can also look at each single nerve. We know each single motor neuron being in this nerve, which gives us an enormous precision as readouts. Secondly, we can culture these cells into primary culture, which gives us full access to the genetic combinations that we can make in the fly, and then test them in the tissues, cell tissue dish. Now, if you look at the cell cultures, then you can see here that these neurons display dynamics. This is the backflow of actin. This is the polymerization of microtubules, which, when tested and comparing them to literature, are very similar to what we know from vertebrate systems. So I think we've got here a fantastic system in which to progress. And I would now like to show you a few examples of how we can conceptually make sense of this very complex picture that I showed you a few slides before. Let's start with cortical actin rings, which will lead over to microtubules, you'll see. So it was published in 2013 in the mouse that axons have these very regular rings of cortical F-actin, which are connected by spectrins. Now, two people in my lab, Yue and Ines, took on the task of looking whether in Drosophila we may find very similar rings, and indeed we found them. You can see here the nice spacing. This was done with structured illumination microscopy. Now, the big advantage in fly was that we could throw genetics at this system. So what factors are required for the formation of these rings was one important question. And we looked at over 15 different conditions. And this was done actually not in Manchester, but we had to travel away. This was done in 3 weeks. The preparations were done in Manchester. The imaging was done in 3 weeks. And we basically could classify that elongating factors of actin played little role, nucleating factors a strong role, and cross-linkage of microtubules also was very important. Now, I don't want to go into detail of actin regulation here, but this gave us a whole set of conditions which we now could test and ask the question of what are the functional requirements of actin rings. And one of the key findings was that if we took actin rings away, we saw defects also. It shows situations in which microtubules in these neurons were already destabilized, but looked fairly normal. And we then took our different conditions, just move this up here, where we have conditions where the actin rings are gone or where the actin rings are fully present. And we then looked at whether microtubules were stable or whether microtubules were lost. And when we did so, we found a very clear and very close correlation. You see here the coefficient between the less actin rings there were, the less stable microtubules were. And this could be driven and this is shown in the next sequence. So we have here a condition where these neurons are polymerizing. You'll see the film in a second. So they have normal polymerization of microtubules in spite of these neurons being deficient for microtubule stabilizing factor. But now we are adding cytokinesin D, which depolymerizes F-actin. And you see that within a matter of minutes, we get a decay of microtubule polymerization. So you see this here. Microtubules are polymerizing normal, we then add cytokinesin D, and polymerization vanishes, and these axons retract. We see this again, normal polymerization, hold of polymerization, and vanishing of polymerization. So we can, in these conditions, destabilize axons and lose axons, a condition that has never been shown anywhere in the literature before. And this shows that actually the combinatorial genetics is a very powerful tool. But now, of course, the key question is, what is the mechanism? And so far, I cannot really tell you the mechanism, which is something to be awaited in the future. But we made progress in a fundamental first question, which is the question of what is the core machinery of microtubule polymerization? So we know that there is polymerization taking place in an axon, but actually there is not a single condition apart from this in which we can abolish this polymerization. So what is the machinery that regulates polymerization in an axon? And we wrote a review about this, which was published in 2016. It's all linked out here. So if you go to my talk later on, we'll make a link available. Then all these papers are free access. And basically, we came up in this review with this picture of polymerization control. So it looks complicated, but there is some simplicity behind this. We have proteins which are localizing to the polymerizing tip of microtubules. We have proteins which are binding along the shaft. And we have proteins which deliver new tubulin for the polymerization process. So we looked at, wrong direction, using the wheel, the mouse. We looked at many different factors involved in this in the fly, asking which factors do we think are really essential, and work by Natalia and Ines primarily, other people got involved later on, came up with these three proteins as key factors. Tau, which is a shaft binding protein, EB1, which binds behind the polymerizing tip, and XMAP, which is called MSPS in Drosophila, mini spindles, which is a polymerase binding to the very tip of microtubule. So why we thought that these proteins are working together in some way is that they displayed the same phenotypes. So first of all three of these factors, when taking out of the fly system, we saw shorter axons. Microtubules became severely disorganized. And if we looked at the polymerizing tip, then normally we'd have stable comets, as we call this, formed by EB1 at the tip. Whereas in all three conditions, these comets became shorter and or less intense. So we lost EB1 from the plus tip of microtubules. Now, what was then also important is that when we looked in vivo, we could find that there were conditions taking these factors out, that this even happens in vivo. So we get disorganization in vivo, showing that there seems to be something relevant here. And if we make combinations of these three conditions, then we could find that there were strong genetic interactions between the three genes. So these three genes seem to work in one common mechanistic path. We collaborated with other people in the field. Now let me put this down here again. So with Paula Slater and Laura N. Lowry, for example, they looked in xenopus for us, so in frogs, and found that actually the same three genes seem to work also in a functional context with very similar readouts in the frog. So something that we discovered in the fly seems to be conserved into vertebrates. Now, I don't want to go into further detail here about the mechanisms, but I want to stay rather conceptual. So basically, we found that MSPS is required to polymerize microtubules. This leads to these yellow dots here, GTP tubulin cap, which is important for EB1 to bind. Whereas EB1 makes sure that the new tubulins are properly aligned, structurally aligned at the tip, which helps MSPS to bind. So we've got here kind of a positive feedback loop between the two. And it is tau along the shaft that makes sure that EB1 binds to the tip and does not bind to the shaft, and that we're maintaining populations of EB1 at the tip. So these are the mechanisms that we came up with, but one of the fundamental questions we still needed to ask is, why do microtubules become disorganized? And this question leads over to the next bigger concept, which is that our 50% of microtubule regulating factors that we looked at in the fly, and here are very different factors. We'll come into all of these factors during the talk. We found that microtubules become disorganized. And if you look at these pictures, then it's very difficult to see distinctions between them, perhaps in quantity. But if I would do double-blind experiments with you and show you different phenotypes, you would be unable to distinguish between all these different conditions. So the point here is, why do microtubules show always the same phenotype, although the genetic conditions are extremely different? So of course, the other point is that these disorganizations here are very similar to what I described to you before in axon degeneration, also in humans or primates. So is this a model that we could use in order to understand this condition? So Andre looked into different conditions with live imaging. You see here that disorganization is being generated. So we can see this over time. And if you plotted this in wild type, that whenever disorganization happened, then basically this was suppressed in the wild type. Whereas in any condition that he looked at, a mutant condition he looked at, he saw that disorganization would never recover. So obviously we are looking here at a condition which is somehow debilitated. So how can this be explained? I wrote a review recently, again this is linked out down here, in which work is described in vitro. And in vitro, if you plate microtubules on kinesin carpets, so kinesin is a motor protein, and these microtubules are now moved around, then these kinesins form rings. And these rings are exactly in the kind of size that we are observing in flies. Now, there are models for this that can be done, and I don't want to go into greater detail here. This can all be read in this review. We believe that it's the kinesins and the high density of kinesins in these axons, which is causing microtubules to go into disorganization. So in axons, we have very similar conditions as in these in vitro experiments. We have a high density of kinesins, we have stabilized microtubules, and a high microtubule density. So therefore, the model goes as follows. We would say that normally you've got axons, and in the axons, microtubules are constantly polymerizing. By default, these microtubules would want to go into disorganization because of kinesins driving them into this condition. However, we have microtubule regulators, and these microtubule regulators are taming microtubules into axonal bundles. Meaning that if we take a microtubule regulator away, this weakens the system and the chance for microtubule disorganization is increasing. Now, one mechanism that we found in this direction is the cross-linkage of polymerizing microtubule tips through a molecule called SHOT, which is a spectra plugin and highly conserved in mammals, which at the same time can link to F-actin in the periphery. And in that way, we've got basically guidance of microtubule polymerization in parallel to the cortex, and therefore laying microtubules out into parallel bundles. Now, if we take SHOT away, then we get this disorganization. You can see here how microtubules are polymerizing off track in this condition. We then looked in mouse for taking the relevant or one of the homologs out in mouse. We found exactly the same phenotypes. And there was a second homologue in mouse called dystonin, which was published already in the 60s, showing that one of its hallmarks is disorganization of microtubules in axons and axon degeneration in the sensory system. And actually in 2012, the same year that we published this paper, a human mutation was shown to cause phenotypes or symptoms which are very similar to degeneration in the mouse. So we think that there is clear relevance. We can unearth concepts in the fly. These concepts may then be able to explain conditions also in humans or mouse models. Now, this is one mechanism, so guiding microtubules in parallel to the actin cortex to lay them out into parallel bundles. Other mechanisms one could imagine is, for example, cross-linking of microtubules, thus basically holding microtubules together. This is a whole area that actually needs to be reinvestigated, and if you read my reviews, then there is many comments about this. So I think it's an area that really needs to be explored in greater detail. But I would like to show you one further mechanism, and this mechanism is basically addressing the question, what if microtubules escape these mechanisms and microtubules go off track anyway? So can the system respond? And yes, the system can respond. We identified a factor called EFA6, which is a cortical collapse factor that sits at the cortex and can eliminate microtubules that leave the microtubule bundle and project to its surface. So basically, EFA6 is expressed throughout neurons. You can see this in cell culture, in the larval, and throughout the adult nervous system. So it is there at any time. And as you can see here in live imaging, these are again EB1 comets in wild type. You see that if comets reach the surface, they always vanish very quickly. If we now do this in a condition where there is no EFA6, so no cortical collapse factor, then now comets suddenly can survive at the surface, at the surface of the axon for much, much longer times. So what may be the consequence of this? So how may this affect morphology? One could think of several things that can happen. On the one hand, we may get microtubule disorganization because these off-track microtubules, which are not in bundles, are not eliminated, hence can now nucleate disorganization. We can have more off-track microtubules along the shaft, which can cause more branching. And at the same time, we can also cause more growth because microtubules in the periphery are more stable. And this is exactly the phenotypes that we found in cell culture as well as in vivo. You can see here, this is just the cell culture data. We have more growth in the absence of EFA6, but if we overexpress EFA6, we have less growth. We have more branching in the absence of EFA6, but less growth if we overexpress EFA6. And we have a gradual increase over time in disorganization and different conditions, which is suppressed if we overexpress EFA6. So indeed, we have three phenotypes. One is disorganization of microtubules, increased axon branching, and increased axon growth. So is this function of EFA6 conserved? First of all, we looked at the structure of EFA6, and we identified this little motif here of only 15 amino acids in the N-terminus. This little motif we call the microtubule elimination domain. And this domain we could show in vitro in collaboration with Claire Friel that it could inhibit microtubule polymerization in vitro. If we overexpress this little motif in axons, we could eliminate whole axons. If we express it in fibroblasts, so in non-neuronal cells, we could eliminate whole neuronal or microtubule networks. So it is a very powerful motif. If you look now in evolution, then C. elegans still has this motive, and indeed C. elegans can collapse microtubules in a very similar way as in Drosophila. But actually in C. elegans, this had been published earlier, the existence of this motive. But if we looked at human or mammalian homologs, then it didn't have this MTED motive. So obviously what we find in Drosophila may not be relevant. However, what we then found in the literature is that conceptually we may be very well on the right path, because in mammals we have a different cortical collapse factor, likewise binding to the cortex and eliminating microtubules at the cortex, which is KIF21A. And the phenotypes that we have with overexpression or underexpression of KIF21A are the same as we have with EFA6, which is additional branching and defects on axon growth. So what we believe again is we can raise conceptual understanding in Drosophila, and this then, although sometimes it may not be conserved at the molecular level, it may be conserved at the conceptual level where simply other proteins have taken over similar functions. Good, so I've shown you now EB1, I've shown you EFA6, I've shown you SHOT, why we find disorganization in these conditions. But this leaves us with a very different condition, which is kinesins. If we take out kinesins, we also get microtubule disorganization. This is surprising. So we say that kinesins are actually one of the key causes for microtubules to get into disorganization, because they're putting stress on these microtubules. If we now take a kinesin out, we would expect that it should cause less harm to microtubules. But what we find is the opposite. We find the same phenotype as we find in all these other genes. So this is confusing. We started to investigate this, and Yuting Liev, PhD student in the lab, took this on, and she basically screened to a whole range of different kinesins. We found that three motor proteins. We found that kinesin 1, kinesin 3, as well as dynein, dynactin motor proteins were causing this disorganization of microtubules. Now, interestingly, these are exactly the three motor proteins that in OMIM are linked out to axon degeneration. So this may be a coincidence. It may be that there is really a conserved common reason, and that we can use Drosophila in order to find explanations. Now, if you look at motor proteins, and especially kinesin 1 has been studied in great detail to this end, then kinesins can either slide microtubules. So this kinesin 1 can also bind microtubules with the C-terminus, N-terminus and C-terminus, and that way slide microtubules against each other. It can transport organelles. It can transport vesicular cargo. And there's more functions in transporting also non-vesicular cargo, which we didn't look into. But there's different conditions which we can test. And basically, and I again want to make a long story very short here, we looked at, for example, Milton and Myro, which are linking out to mitochondria. And you see that Milton here also causes severe microtubule disorganization, as does KHC, so kinesin itself. If we look at cargo transport and links, then we have KLC linking out to cargo here. And if we take that out, we also find disorganization of microtubules. Now we went through long loops and tried to understand what is the mechanism behind this. And Yuting decided to look into reactive oxygen species, so into oxidative stress in these cells. And basically she found that oxidative stress could induce also microtubule disorganization. It's not shown here. But if we treated our different kinesins with Trolox, which is a factor that sequesters radicals and takes away oxidative stress, then we could basically rescue the phenotype of Milton. So this factor here of KHC was clearly suppressed. Kinesin 3 also but not KLC. So obviously we are looking at different factors on different mechanisms downstream of kinesins. This factor here, we have no understanding yet, we have ideas. Here we know that ROS has to be involved, so a clear direction of research now. But what it also shows us that we are looking at something that has to do with physiology of the axon. So obviously a function that is downstream of kinesins. So all this sounds very confusing, but I will now bring you to the last slides in which I will explain our fundamental model in which we will bring all these different experiments together. So what conclusions can we draw? Well, I showed you this nice, very complex picture. I showed you then that in this organization, we have looked already at quite a number of factors and have shown phenotypes for these and mechanisms through which they operate. But now can we put things together so that we can get a true conceptual understanding of axon biology? And this brings us to our model of local axon homeostasis, or as we would call it now, the dependency cycle of local axon homeostasis. Simple is the system is very simple. So if you think of axonal transport, then this is absolutely essential to provide materials, components, and physiology, which then can drive axonal function. Any interruption of axonal transport will be harmful to axonal function. Now, in order to perform this axonal transport, we require microtubule bundles because the axonal transport uses them as highway for motor proteins. Therefore, microtubule bundles are absolutely essential. But at the same time, I told you that axonal transport damages microtubule bundles. So it drives them into curling, for example. Now, this requires that we need mechanisms that can maintain microtubule bundles. And I showed you a number of them, like, for example, polymerization linking out to a shot. So polymerization establishes EB1 at plus tips that links shot, and shot can then link up to the cortex that guides microtubules into stable bundles. EFA6 can eliminate microtubules that are going off track. So we have got a number of maintenance factors, and many more will be found in future, that can counteract this damage which is caused by axonal transport. But now, of course, this maintenance is depending on axonal transport because we have to bring in the components like shots, like EFA6 into the axon. But we also have to deliver physiology. And part of that physiology is, for example, to bring the right species of reactive oxygen species into the axon to prevent oxidative stress, but also to bring in ATP, which is required for transport, and so on. So we've got also internal loops here. But basically what we have now is a circle of dependency. So microtubule bundles drive transport. Transport damages these bundles. These bundles can be maintained by using what is brought by axonal transport. And all this circle can drive axonal function. This now means that from wherever we are bringing on, and actually there's also with our what to mention, of course, we also have to maintain the cortex, which we have shown is important to maintain microtubule bundles. And there is now also additional insights published over the last few years showing that the cortex is enormously important for maintaining axonal structure. So what this means that we can now, through genetic defects, stress, aging, trauma, in different positions of this circle interfere, but we will always break exactly the same cycle. This could explain why actually interfering in different positions can cause very similar axon pathology. So if you think of spastic paraplegia, we can take out spastin, which normally acts through this microtubule binding domain in that context, so microtubule regulator, or we can take out ER components, and both of them lead to a very similar disease, which is called spastic paraplegia. All this becomes explainable through this circle. We can explain why, for example, Alzheimer models can be improved by using microtubule stabilizing factors. Good. So hopefully you have seen from this that concepts can be worked out in the fly. And maybe you have become tempted a little bit to use fly for your own research. And this brings me to the last part. So we have made many different resources available to the community, and one of them is this genetic training package. So if you click this link, you can find all the materials required to learn Drosophila genetics in an easy way, which is used worldwide, to establish it in your own research lab. And of course, we are always happy to help. We also established Drosophila as an outreach resource for schools, for example, driving fundamental biology. But this is only our side of things, so that's the Manchester Fly facility. It is Firzan, whom we collaborated with, who founded Fly Indonesia. And as you can see here, many of the resources that we made available for Man Fly facility has been translated also into Indonesian. It's all available on that webpage. Again, the link is being provided, including also our little movie. And this is where I would like to end up, to briefly play this little movie. Oops, let me bring this up, play this little movie to you for a few minutes. Can you hear this? Can one hear this?
Actually, the sound is not working probably.
Okay, because it's just going through the. So basically, you've got the whole film explaining why Drosophila and how Drosophila came into science, all explained in Indonesian. So all these things are available to you. This leaves me with thanking to all the members of my group, whom hopefully I've correctly indicated already throughout the talk, our collaborators as well as our funders. And yeah, thanks for your attention. And hopefully this was a good introduction to how fly can be used to study biology. Thank you very much.
Thank you, Andreas. Let's give applause to Professor Andreas Prokop to give us great presentation about axon biology, the maintenance and how microtubules involve in the physiology of axons. And then maybe it's, I'm sorry, Andreas, it's not my field of study actually. I mean, innate immunity, but I will try to summarize your talk a little bit. Okay, please. Let me know if I'm wrong. Okay, great. So Professor Andreas Prokop has shown us using Drosophila that microtubules play significant role throughout axonal life. And this is really important since no one ever shown it previously using higher eukaryotes. So by using fly, Professor Andreas Prokop's and team show us how microtubules function in the physiology of in the maintenance, in the axonal function. And the last slide of the summary is really good because I finally understand like how everything is connected, like how EFA1 and everything, kinesin 1, kinesin 2, and everything is connected. And I have asked Professor Andreas Prokop to provide us with the presentation materials, and I think you give your permission, right, Andreas?
Yeah, that's fine.
Great. Many people already asked me by private actually because they love your animation. I mean, if I'm not mistaken, you make by yourself, isn't it? This
Fun? Yeah,
this
yeah,
kind
yeah.
of picture.
Yeah.
Wow. Yeah. So what I'm thinking is, Professor Andreas. Sorry, not I'm thinking, but based on the Professor Andreas explanation, that using flies we can do many things in axonal biology with cheaper, easier, and many easier way, and many genetic tools are amenable. I mean,
No, it's
amenable
not only
genetic
that.
tools.
I think what's essential is that we can work out with high efficiency many different factors in the same system. We can generate double, triple mutants, and that is when we normally see the most important phenotypes. If you would want to do that in the mouse, this would take years. And this now with CRISPR, perhaps you can advance a little bit faster. But of course, in fly we also can use CRISPR. So this makes our work even faster. So the key point is really that we did all this work and we looked at over 50 factors in one standardized model, where whenever we find a phenotype, we already know which factors have caused us similar phenotypes. We already know in which direction to look, which is an unprecedented database for this kind of work, and also then to advance it at high speed.
Thank you, Andreas. That helps me a lot. Okay. By the way, I would like to say to everyone. I already said to everyone that if they have question, they can give me like notification in the chat that they want to ask, and already one person want to ask. But I just want to ask you something that is really important, and this is asked by many people already in Indonesia. That do you require ethical clearance to perform fly research in Manchester?
No, this is what I try to explain in my advantage slide. So we don't need any licenses, which is why whenever you have an idea, you can drive your research immediately. Very often fly stocks are already available. You may even keep them in your laboratory already. So combinations of those are generated within a matter of weeks. So you can make double mutants or triple mutants. Sometimes it may take you 2 or 3 months, but not years as in mouse. And it is therefore extremely fast to play around. And biology, I always say, is complex. So in order to unravel complex networks of function, you want to be able to play around and to make errors. So if in a mouse model I decide to generate a mutant, then this is a decision which has lots of implications. People have to work on it for a long time. If this then leads to nowhere, this is a huge problem, also career problem. Whereas in Drosophila, if something doesn't work out, you say, well, fine, I try something different. The time that I've invested, the money that I've invested was fairly minor. I mean, it is still not nice not to get anywhere, but it doesn't kill my career. And these are aspects that also need to be considered. Some of my people in the lab have hobby projects where they say on the side, I'm trying something that I hypothesize could work. And sometimes these areas of research suddenly develop into a main area of research, which become far more revealing than what one originally wanted to do. So this playing around with biology at low risk and low cost is, I think, what is intellectually extremely pleasing and can explain why you can work out concepts so much more efficiently.
Great, thank you, Andreas. And that maybe answer many questions about the ethical clearance in here. Okay, great. One person want to ask. Maybe I will let Miss Bese Hardianti to ask the question. Miss Bese, are you here? Mungkin Bu Bese bisa. Sorry, Andreas. I will ask. I will talk in Indonesian language.
Mungkin
Bu Bese. Thank you so much.
Okay.
Hello, Mr. Andreas. Hello.
Yes.
Good morning. Yeah.
Good morning.
Yeah, thank you very much for the chance for your presentation. It's very nice to know new knowledge about Drosophila related to the microtubules. I want to ask a question, maybe a little bit different with what you explained about. In your presentation you mentioned about kinesin 1. So I really want to know how that the loss of kinesin 1 could increase the ROS expression. So could you explain a little bit about the upstream and the downstream protein that involved in that mechanism, and how to maintain that level of kinesin 1, especially to maintain to stay homeostatic conditions, especially the in the action.
Yes. So, I mean, first of all, kinesin 1 and kinesin 3, partly kinesin 2, are the key transport anterograde transport proteins in axons. And this is true across the whole animal kingdom. Retrograde transport is driven by dynein. So these are the key factors that can bring stuff into the axon or take it out. In order to do this, they require microtubule bundles. So if you would damage microtubule bundles, you would automatically affect axonal transport. Therefore, you would break that transport necessity for the axon and affect the axon. Now, these kinesins themselves, for example, regulate the distribution of mitochondria across the axon, meaning that the distribution of ATP is dependent on these kinesins. The distribution of other organelles like peroxisomes and mitochondria are collaborating in ROS regulation. And of course, there's ROS species which are important, which are helpful, others which are damaging. So basically disrupting the system may cause oxidative stress, and that oxidative stress is something that we observe, which then damages microtubule bundles. So the problem of looking at, for example, kinesin mutants is that you're having effects in each and every direction. On the one hand, these kinesins are, by running along microtubules, cause tear and wear of microtubules. They damage microtubules, so they're harmful to microtubule bundles. On the other hand, they are required to bring in the physiology and the materials that are required to maintain these microtubule bundles. And as such, these kinesins are upstream, downstream of microtubules in damaging them as well as maintaining them. So it's a very complicated system. And I think we are making some progress in trying to understand that. The ROS is a very helpful first angle, and also collaborators in Liverpool are making huge advance in trying to understand how ROS, for example, impacts microtubule behaviors. Does that answer your question?
Yes, that's very nice. Thank you very much for the answer.
You're welcome.
Thank you, Andreas. Okay, great. The second question will be from Miss Yulia Jabir, PhD. So please, Miss Yulia.
Okay, thank you, Mr. Firsan. So hello, Professor Andreas Prokop again.
Hello.
Yeah, I have a question about, do you ever try to make a Parkinson disease model in Drosophila animals?
What disease model? I didn't understand the word before.
Parkinson diseases. Parkinson disease.
I mean, we personally not, but there are existing Parkinson disease models. And again, I mean, it's something that if you want to work with Drosophila, I always tell people, and this is true, whoever works on it, first become a specialist in your area and understand exactly what are the key gaps in the field, and then ask yourself, how can you use Drosophila in order to address those gaps? And that will really advance the field. So this really requires that you very well understand the field and have the background knowledge, and also understand how Drosophila is being used already. In this context, you have a number of proteins, like, for example, Parkin, but also, what's the name of the protein, which is involved also in mitochondrial transport. Now I don't get the name at the moment, but
is it pink?
It's Alex Whitworth, for example. If you check up that name, he has worked on Parkinson Drosophila models for many years. So there is examples where Drosophila has been used to understand fundamental functions of proteins linked to Parkinson, and this understanding then was later on shown to indeed be relevant in the disease. So one of the proteins was pink.
Okay.
Pink, Parkin. So there's a number of proteins which have been looked at in Drosophila and studied in Drosophila. And most of that, to my knowledge, is relevant then to the actual understanding of the disease.
Okay, thank you.
You're welcome.
So Andreas, actually Miss Yulia Jeber, PhD, was worked with Parkinson disease when she did her master degree, I think.
Yeah.
Yes.
So maybe that's why she asked that question.
So on what model did you work? In which model did you work?
We used mice.
Okay, so I mean, it's of course the fundamental question. Parkinson is a complex thing, and one doesn't really know why degeneration starts there. There's all these links potentially even to the enteric nervous system, and obviously it's something which starts in one area preliminary. And it is quite interesting. There were papers published already in 2000 that if you overexpress, what was it? Forgot the name at the moment of that. So basically, they took a disease-linked version of one of the Parkinson-causing genes. Overexpressing them in the nervous system led to preferential death of dopaminergic neurons.
Yep.
It was a paper by Mel Feeney and
Bender
in 2000.
Yes, it's Feeney and Bender in Nature.
Yeah.
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 Great.
So we did try to. I mean, I tried to ask Mr. Fearson before that if we can like develop this model in Drosophila, and that would be great if he could help me with that.
I would advise you contact Alex Whitworth, who is one of the leaders in that area in Drosophila, and he will tell you very, very clearly what's possible. And if you have concrete questions as to what kind of questions you want to address, you could discuss with him what may be ways forward. And as said, if you want to establish Drosophila in your laboratory, it is easier than you think. And if you go to the link that I provided about the Drosophila training, this training has been downloaded about 30,000 times across the world. Many positive feedback. So it basically explains from scratch, why do we use Drosophila? What is happening in the laboratory? What are we crossing in fly genetics? And how do Mendelian rules apply? How do we keep flies? And so on. So it basically gives you a very from scratch introduction to fly, and it's a manual of about 30 pages. So that gives you a first impression as to what will happen and what is possible.
Wow. Great. Thank you, Andreas.
You're welcome.
Thank you for the questions. Okay. Is there any other person who wants to ask question? Please tell me whether. Sorry, Andres, I will talk in Indonesian a little bit.
That's fine.
Oke. Kalau misalnya ada yang ingin bertanya bisa sekalian langsung, atau kalau misalnya ada yang ingin bertanya dalam bahasa Indonesia bisa diucapkan saja, nanti kita bantu translatekan. Bisa raise hand atau misalnya kasih informasi di chat bahwa ingin bertanya. Okay, great. That's basically information about if someone who wants to ask question.
Yeah, understood.
Yeah, Andreas knows a lot of Indonesian language now.
You even offered to translate, I understood.
You don't have to translate, you already understand. Andreas, while waiting for the question, I have one question actually related to Trollocs. You use Trollocs in the experiment, isn't it?
Yes.
Related to Ross. What is?
Yes.
Can you explain a little bit more? What is Trolloc and how do you use it in your research? Like, how do you use it in technical things, like putting in something?
Trollocs is potentially vitamin C, which sequesters radicals. So one of the reasons why it is also helpful to eat vitamin C. Or is it vitamin C or vitamin E? I forgot now. So basically all you do is you add Trollocs to your medium, or you can also feed it to flies, and you can then see improvements of the phenotypes. It is very straightforward.
Do you use it in vivo? Like, I mean, in the adult fly?
You can feed it to flies, yes.
Oh, that's wonderful. Yeah. Okay. Another important question is like, what is the clinical? I mean, actually, you already explained and summarized the function of the physiological function of this empty microtubules in the axonal biology. But do you have any idea of the clinical relevance of this empty disorganization? Like, you already presented several types of disease linked to the this Axon biology, but have you tried to discuss this thing to, for example, medical doctor or something?
So one of the key problems, and this is in our review from 2019, about the model of local axon homeostasis, which does not yet have the circle, because at that point we hadn't understood the circle yet. The circle is something that came very recently and now actually makes even more sense. So everything that is written in the review makes sense to me, but it makes even more sense now when considering the circle for the cycle. So in that review, and this has about over 400 references. I really went through the literature very detailed to find out what we know in axon pathology about microtubules. One of the key problems that we are having in the field is that microtubules are very rarely looked at. So the majority of studies when doing electron microscopy looks at mislocalized organelles, but doesn't go to the resolution of microtubules. So in those images, there's first no mentioning about microtubules, and also in the images, the resolution is too low to say what may be going on with microtubules. The second point is that in cell culture, people are using much more neurofilament antibodies rather than microtubule or tubulin antibodies. So again, in those images, even if it is not the focus of the paper, we cannot even in between the line extract information. So I think this is one of the first messages to anybody working in pathology. Also look at microtubules. So this is one of the messages we need to bring across. In that review, I've listed all the kind of diseases where we have some knowledge, and there is, I think, good support that microtubules are linking to pathology. So I've given you in the talk a number of examples. One of them was the dystonin. where we have human proof that if you interfere with the microtubule binding of dystonin, which is the linker to the actin cortex, then this causes a very similar phenotype as we find, which is microtubule disorganization. It's one of the strongest phenotypes in mouse, as well as in Drosophila. If you look at EFA6, then we have cortical collapse. This taking out the cortical collapse factor, in that case KIF21A, which is not the same as in Drosophila, but is playing very similar roles, you get a disease, which is a dystrophy of nerves that project towards the eye. And this dystrophy seems to be due to very similar mechanisms as we discovered for EFA6. So overall, it is really to by and by build a stronger foundation so that people buy into these ideas. I think it is at the moment really opening up your eyes towards microtubules, include them in your research, deliver more descriptions in pathology, what is going wrong, and then seeing and testing whether the model may explain these kind of aspects or to which degree we have to refine and improve the model.
Great, thank you, Andreas. Basically, there's some question here. Two more questions. Are you able to?
Yeah. There's also in the chat. There's something going on in the chat, but it's in
Indonesian, yes, in the chat. I will read in the chat if you don't mind. Okay, Professor Andreas, did variety of food affect growth and development of axon?
Food?
Yes.
I would not have direct evidence for that. So, I mean, of course, food and nutrition is being studied in Drosophila. It is being studied in various contexts. So, for example, the impact of food on neural development and also on behavior, on lifespan is being looked at intensely. And also the microbiome in Drosophila is perhaps one of the best studied microbiomes we have, because the number of species of bacteria living in the gut of Drosophila is far lower. We have very good genomic information about them. So it is really a network and a system which can be much better understood if we really want to unearth and understand the fundamental concepts, how the gut biome influences other aspects of physiology. So that's all I could say about food.
Yep. Thank you, Andreas. Another question from the same person, I think, is: did your research with Drosophila can contribute in the field of neurology, like Alzheimer or another genetic disease?
Yeah, that's what I try to explain briefly, and perhaps if you re-look at the talk, then these things become clearer. So it is one of the key questions in Alzheimer's is really why nerve cells die or get into pathological condition. And this is a question that, again, requires systemic understanding. And that is where the kind of cell biology that we are driving is becoming extremely helpful. Because you can, like, for example, our cycle. If you think of Alzheimer models in mouse, for example, can be treated with epithelion B and improved. And epithelion B is a stabilizer of microtubules. Now, if you think of our cycle and dependency cycle, that let's say you affect in Alzheimer's, you have a certain condition which affects the physiology of an axon, then why can we rescue this by stabilizing microtubules? The cycle starts making this clearer. And since Alzheimer's is a multifactorial disease, I think we cannot come with one explanation, but our cycle, for example, can deliver explanations as to how very different causes can lead to very similar phenotypes and symptoms. So that's, I think, where our model can be very helpful. And of course, the different aspects of the model, of the cycle, and I hopefully have given you examples, can be studied in the fly. Once they are understood there, one can then take that knowledge and test similar things are true in the fly, in the mouse. And we've done this already. Like, for example, looking for whether taking out spectral plugins like shortstop, dystonin, ACF7 causes similar phenotypes in mouse, and it does. And we have a number of examples. Or if you think of Xenopus, where we can show that the same 3 proteins, which are collaborating in polymerization in Drosophila, seem to do the same in Xenopus.
Yep. I saw in the presentation about Xenopus. Yes, indeed. And then, this is Indonesian question. I think the last one. Okay, the last one is: have you ever used other model systems other than Drosophila?
So we have established in the lab, we had established for a while also work on mouse. But I've taken the very clear decision because we have are surrounded by many other groups. So I've decided that I mainly want to do work in other models through collaboration, because maintaining expertise in the lab is costly and is stress factor. And I'm just convinced that the kind of questions I want to ask can best be addressed in the fly at the moment. So it is the points where we have the feeling we understand something or have understood something new, where we would then think about whom can we collaborate with. So I'm myself not maintaining any other model system in the lab, but I always have my eyes out there to think about where and whom could I collaborate with in order to bring this relevance to our findings.
Great. Yes. I got you now. Because we in Indonesia mostly work with mice, rats, and like sometimes rabbits. So
yeah, as I showed you, I mean, just looking, I mean, the costs in Indonesia may not be as high as here in England. I don't know, but I guess maintaining mice is a huge drain on your finances. And in Drosophila, with Drosophila, really you have 100% cost savings. I mean, I've given you that example of maintaining. I mean, if you would want to maintain, of course, you can deep freeze your embryos in mouse. But of course, that means also that to defreeze them and get them accessible again as a mouse train takes quite some considerable time. So it slows you down in your progress. So the advantages in being able to do that, but also they are not research ready for you. Whereas every fly stock we are keeping is research ready to us, and it costs me nothing. It costs me £100. And I guess, again, in Indonesia, this would be cheaper because, for example, paying a student or cooking the food, all these things can be arranged in ways that are not very costly at all.
Yeah, I can say to you that we spent so few amount of money to maintain Drosophila, actually. And now I bring some of Drosophila to my house because the COVID-19 pandemic requires us not to use the lab for some time at that time. So yeah, so I cook the food by myself, and just, it's so maybe I can say maybe £1 for something I can use for some time for fry food. Yeah, that's so cheap. But the problem is, Andreas, I can tell you that if we order fly stocks from stock centers, it takes ages, like to go to Indonesia. For example, when you sent me fly,
this is where your collaboration with India, for example, could be very helpful, and building infrastructure in that direction. It all depends on building momentum, and I guess once there is a number of people who are doing this, one can establish links to close stock centers where one has an established route in which things can be sent much quicker, and then you can distribute within the country using your knowledge about the Indonesian infrastructure, and of course also the problems and how to overcome them. So all this can be arranged, but it requires a community to come together and build that infrastructure.
Yes, I agree with you. That's why I'm trying to, for example, taking some stocks from Japan, from Singapore, and then I can distribute them when someone in Indonesia require. Because, for example, if I order from you in UK, it takes 2 months to come to Indonesia. And when I order fly from BDSC, Bloomington, it took 3 months. Like all of them died.
I'm also supporting Joseph from Nigeria. They have very similar problems, but they have talked to Kevin at the Bloomington Stock Center, and basically the idea again is that Kevin has tricks to send these things. If one has a firmly established link, then one can smoothen that process, and then at least they are already in the country. Then one would have to make sure that they are maintained. For example, if you would be such a hub, you would have to maintain them until they have safely arrived at another person in Indonesia. So these things, I think, can be built. And then you also have the links to Dipti in Bangalore. So India may be another possibility to build such a hub. It's really all about being inventive, creative, and building an infrastructure that works much smoother than the occasional activity that you're doing at the moment in this direction.
Thank you, Andreas. I believe your saying is heard by our dean and vice deans. Maybe they can help me to build kind of like how to say little stock center for Indonesian universities. And we can say that Unhas is the in collaboration with you or other institutions in the world, try to bring this Drosophila into Indonesia. Okay.
I mean, we are also with Drosophila Nigeria, for example. At the moment, I'm very busy, but hopefully in a few weeks we've uploaded materials about how to keep flies under different conditions. I mean, in Nigeria, often have the problem that there is, electric faults all the time. There is the conditions are just not very good. You have to be inventive as to how to deal with your flies, and there's lots of little tricks how to sleep your flies. By using sodium carbonate, you can make CO2 on the spot, to sleep your flies. So there's many tricks you can use, and those things will be on the webpage in due course and can then expand from there. And it's all about, for example, using Fly Indonesia as a webpage in order to collate common knowledge, right? So if somebody comes up with new tricks or new ideas, to share this knowledge on a common platform where then the community can stabilize itself and can increase its momentum by basically no longer making it necessary to reinvent the wheel, but to dip into existing tips and tricks, and therefore be able to apply them much quicker and establish flies much quicker.
Great. Thank you, Andreas, for your answers. And maybe this is the time for Mr. Muhammad Nur Amir. Maybe you can explain about the link that is sent into the group chat. Pak Nur Amir, bisa dijelaskan sedikit Pak, itu link apa Pak? Bahasa Indonesia aja Pak.
Iya, sebagai daftar hadir buat peserta yang ikut. Jadi nanti
Enggak kedengaran suaranya Pak Nur Amir. Mungkin karena pakai ini headset kali ya.
Ya. Jadi link pendaftaran yang telah kami kirim itu silakan Bapak Ibu mengisi nama lengkap dan email yang aktif. kedepannya nanti akan sertifikatnya akan kami kirimkan langsung ke email Bapak Ibu peserta. Tapi tolong jangan di-share keluar karena tadi jumlah peserta maksimal yang hadir itu cuma 176.
Yeah, yeah.
There was just another note in the chat.
Yeah. There's a question actually. Do you mind to answer that?
No, it's fine.
Okay. I will read in English this question. Do you think if we do experiments using Drosophila, this is related to drug discovery actually, Andreas?
Yeah.
Can we obtain the results faster and effective compared to other model organisms like rats, mice, or something like that?
I mean, you are in a much better position to answer that question quickly because that's what you do. But of course, I mean, I know that, for example, in my neighborhood, Richard Baines is working with epilepsy, and they systematically screen through drugs, drug candidates in flies, can do this in large amounts. They have done this in collaboration also with clinics, where basically some tests that were found in Drosophila were then tested in the clinics. So there are fantastic possibilities. I mean, the best example that I'm aware of is epilepsy, and basically we know that epilepsy is caused by, I mean, the epilepsy that relates to channel proteins or to action potentials. We know that the same drugs, the same channels are affected in humans as in flies, and you can use the same drugs that are known to work in humans, and they will improve the condition in flies. So there's a strong case to be made. Of course, one always has to be careful. It is only a pre-screen, I guess, you would do in Drosophila to limit down to further candidates. But sorry, I think you're in a much better position. I think many people...
Sorry, Andres, someone is sharing screen, sorry. I will notify him. Zulfi Ayu, tolong di ini apa? Oh, sorry. Yeah. Okay. Tolong host ini apa? Disable dulu untuk sharing screen. Yeah, please, Andreas.
Okay.
Yes, so I mean, the answer is in Drosophila, I think you can do it much more efficiently. And again, just think of the numbers. I mean, the numbers of rats or of mice you would have to use, the time that you have to spend in flies. These things can be done. I mean, if you just think, for example, if you want to look into aging and drugs that concern aging, a colleague of mine in Liverpool works on aging, also on microtubules. So many of the findings that we have on microtubules are relevant in aging. And she basically can find typical phenotypes, aging hallmarks in the brain already after 5 weeks, meaning that if you want to work on a drug that concerns aging, you can do your experiments in a matter of 6 weeks, where in a mouse or rat, you would have to wait 15 months. So for a pre-screen, of course, this is fantastic.
Yeah, that's really a good example, Andres. Thank you. Actually, the one who asked this question is the head of the postgraduate program in the Faculty of Pharmacy. And then, that will be a lovely question and answers for everyone who is interested to do drug screen using Drosophila. Yeah. Thank you, Andreas. Maybe there's no other question anymore. And maybe we can say thank you very much from everyone in here. Yeah, we can clap our hand. Maybe if it's not here, it's okay.
Thank you very much.
Thank you, Andreas. It's really lovely to see your presentation and also great images and animations. And you already said that we can have that animations. And I will upload
share the link.
Share to everyone who wants to have that PowerPoint file is okay, or should I put
PDF.
into PDF file?
Good. So I mean, you can share the link. That's fine.
Oh, okay.
Thank you very much.
Thank you very
Good,
much.
so, yeah, for me, it was also a very interesting experience, I must say, because talking to a computer is not the same as talking to an audience. And of course,
Yes.
not seeing any responses anywhere is very difficult. So it was an interesting experience, but I enjoyed it. And also thanks for the discussion. And hopefully, I mean, my key interest is I didn't want to overload you with science. But rather try to show you how you can make conceptual advance even in this small organism. Still reminding that we have 10 Nobel laureates who gained the Nobel Prize, so it's 6 Nobel Prizes, which were gained through work in Drosophila, showing you the relevance of this. So there's huge opportunities. One should not close one's eyes towards these opportunities. And I mean, I argue the same in England, and I sometimes get grant applications where people want to do fundamental biology in a mouse, where I would just ask myself, as a panel member, would I not ask these people to first try it in a simpler organism like C. elegans or yeast or Drosophila, or sometimes zebrafish, but zebrafish already is much more complex. To get fundamental answers and then to do translational work into mouse. This would save us a lot of time. It would drive the science to higher quality, and it would allow us to really advance in our fundamental questions while saving a lot of money. So one can only say these things. People have to think about them. We have also lots of outreach materials. If you go to our webpage, like Joseph for Schools, for example, Why the Fly, there's lots of links there that can be used that explain the situation in greater depth, give examples. Just convince yourself as to whether it would be something. Become an expert in your area to then address the question as to what could be done in Drosophila to address the key questions that need to be asked. And is it possible? Of course, there's limitations. Drosophila can't do everything. Drosophila can only do things that are relating to fundamental biology. And also there are limitations. So if you think of skeleton, the human skeleton, of course, flies don't have bones. But
Yes.
if you want to work on extracellular matrix, which bones are, then of course Drosophila has extracellular matrix. So fundamental concepts of extracellular matrix can be looked at in fly. So it's really being clear about what is possible, where are the limitations, and where could the fly be a fantastic tool in order to drive my scientific interest.
Yeah, that's correct. I definitely agree with you. Similar to immune system, it is difficult because we cannot study adaptive immune system in Drosophila.
Exactly.
Yeah. Cool.
But then we've got a Nobel Prize which was on innate immunity, which was basically building on work in Drosophila. So yeah, there is possibilities one wouldn't imagine.
Actually, Andreas, I just want to inform you that next week we will have, inshallah, a presenter from University of Oxford, Professor Petros Ligoksigakis. He studied under Jules Hoffman's lab. I mean, in Jules Hoffman's lab as a postdoc. So maybe he can share us that kind of information in the future, in the next week.
Fantastic.
Good. Thank you.
It was a pleasure.
And one more. Sorry.
To you, I have to say good night.
Sorry, Andreas. Can I ask one favor of you? Can you choose one question that you think the best for you? Because we provide a present for. Sorry, I forgot to tell you that we provide 2 presents. I will choose one, and you can choose one. That question that you think is best to give present. Do you understand what I mean?
No.
So sorry. So we are. I mean, many people already ask questions to you, isn't it? And then among those questions, please choose one question that you think is good, and then we will provide a present.
Okay.
Yes. Do you remember the question? Like, for example, the first lady asked about.
So I mean, my two top questions would be the kinesin one.
Okay.
And the one on drug screening.
Oh, okay.
So you cannot tip a coin as to who wins.
So okay, in that matter, I will give both of them because I don't have to choose. It's easier for me. Great. Yeah. So kinesin one is, I just want to make sure, is from Miss Besse Hardianti, isn't it? Oh no.
Yeah,
Kinesin.
Mrs. Besse.
Mrs. Besse. Yes. Okay, great. So two persons who got the present, which is I will say it later, is Miss Besse Hardianti and Latifah Rahman, PhD. I mean, Dr. Latifah Rahman for the question. Great questions. Thank you very much, Andreas.
You're welcome.
Yeah. So
bye-bye.
This is the finish then. Bye-bye, Andreas.
Thank you very
much for your
Thank you very much,
Bye-bye, Andreas.
Mr. Andreas.
Thank you. I hope please take care. Okay. Ya, oke. Terima kasih semuanya. Jadi presentasi dari Prof. Andreas sudah selesai. Dua orang yang mendapatkan hadiah dari kami adalah Ibu Dr. Latifah Rahman dan Ibu Besse Hardianti. Insya Allah sebentar saya hubungi untuk memberikan hadiahnya. Mohon maaf
juga,
cuma
host juga,
host. Nanti host Bu Yuli yang kasih.
Iya, jadi tolong diisi tadi daftar hadirnya karena itu penting sebagai data kami untuk memberikan sertifikat.
Terima kasih panitia.
Terima kasih Bu Besse. Jadi, terima kasih atas kehadiran Bapak Ibu dosen dari Fakultas Farmasi.
Gimana caranya isi?
Bagaimana caranya isi tuh Pak Nure? Jelaskan sedikit.
Diklik saja Pak, diklik saja.
Diklik.
Google Form nanti itu Pak, nanti bisa diisi nama.
Saya coba share